name is Deborah Park. I'm a 6-year PhD candidate in the Pharmacology and Toxicology graduate group at UC Davis. I work at Dr. Karen Zito's lab where we focus on the molecular and cellular mechanism that's thought to underlie learning and memory formation. This amazing ability for us to learn from past experiences depends on our brain to undergo dynamic rewiring of its neural circuitry. The structural changes required for learning and memory formation has been well observed in animals, where we can take a look into its brain. If we were to zoom in, we would find that brain cells or neurons have these arms or branches, and they're studded with these small bulbous protrusions called dendritic spines. They're sites at which a lot of communication between neurons occur. They're very dynamic. They can either grow and persist over time, or conversely shrink and disappear. Both of these processes are important for learning and memory formation, and also very well regulated throughout our lifetime, so that most of us follow the stereotypic trajectory of spine density throughout our life, as indicated by the black line. Any deviations from this is associated with neurological or psychiatric disorders, one of which I'll be focusing on is schizophrenia. As you can see by the green line, schizophrenia is associated with decreased spine density, which has been observed through postmortem stainings of patients. Another observation associated with schizophrenia is decreased levels of d-serine. This is a naturally occurring chemical that's found in our body that, along with glutamate, interacts with a protein called NMDAR receptors that are present on the dendritic spines. With glutamate, it assists in the function and opening of NMDAR receptors that is crucial for both growth and shrinkage of dendritic spines. So we were thinking that the decreased levels of d-serine in people with schizophrenia alters NMDAR receptor function that then contributes to the loss of spines. To kind of more go into detail of what may be exactly happening is a schematic, where on the left we have what may be going on in people with any psychiatric disorders, where you have both glutamate and d-serine that combine to allow for both calcium and something called non-ionotropic signaling. Both of these are important for growth and stabilization of spines. However, in people with schizophrenia, the decreased levels of d-serine but uninterrupted binding of glutamate blocks calcium but still allows for non-ionotropic signaling, which by itself leads to shrinkage and blocks growth. To test this novel hypothesis, I am using a schizophrenia mouse model that cannot produce d-serine. You can find out some of my exciting results in my poster, and hopefully my study will help further our understanding of how schizophrenia develops and potentially contribute to development of new therapeutic options for this disorder. Thank you for listening, and thank you to the ARCS Foundation for supporting my research.